All right, welcome. As future clinicians, getting a solid grip on the cell's command center is, well, it's absolutely fundamental. See, the genes tucked away in the nucleus don't just handle heredity. They are the master conductors, orchestrating the day-to-day life of every single cell in our bodies. So let's dive right into the genetic control of our cells.
You know, this is one of the biggest questions in all of medicine. Think about it. How does a heart cell know to be a heart cell and a kidney cell know to be a kidney cell when they have the exact same instruction manual? The answer, and it's a beautiful one, lies in this incredibly elegant process called gene expression.
So here's our game plan. We're going to follow the information from the top down. We'll start with the DNA blueprint, see how that becomes an RNA message, and then watch how that message is used to build all the cell's machinery. After that, we'll look at how this whole process is controlled, and finally, what happens clinically when things go wrong. Okay, first stop, the library of life itself, locked away securely inside the cell's nucleus. This is ground zero. It's where the instructions for absolutely everything the cell is and everything it will ever do are stored. Isn't it just wild to think that our entire immense biological complexity is run by only about 20 to 25,000 protein coding genes? So how in the world do we get over 100,000 different proteins from that? Well, one of the cell's cleverest tricks is something called alternative splicing. It's like taking a single recipe and then cutting and pasting the instructions in different ways to make several distinct dishes. It is a fantastic example of genetic efficiency. So these are the basic nuts and bolts of our genetic code. You've got the phosphoric acid and the deoxyribose sugar. They link up to form the two spiraling backbones of the DNA molecule. Then, in the middle, you have the bases pairing up. A always with T and G always with C. They act like the rungs of a ladder, connecting the two strands and creating that iconic double helix shape. And here's the secret to the language the cell uses. It doesn't read one letter at a time. No, it reads in three base words, which we call triplets. So a sequence like GGC, for instance, is the code word for the amino acid glycine. The specific order of these triplets is everything. It dictates the exact sequence of amino acids, and that in turn determines what the final protein looks like and what it does. Okay, so here's the critical question. How does the cell get these incredibly precious instructions out of the fortified nucleus and into the cytoplasm, where all the action happens? Well, it does it using a middleman, a messenger molecule, in a process called transcription. This two-step flow of information right here is the central dogma of molecular biology. It's the core principle. First, inside the nucleus, DNA is transcribed into a mobile RNA message. Then, that message travels out into the cytoplasm, where it's translated into a working protein. It's how the cell turns the blueprint into action. Now, check out the key differences here, because this is what makes RNA the perfect mobile messenger. It uses a slightly different sugar, ribose, and it swaps out the base thymine for uracil. But the big one, it's single-stranded. That makes it the perfect temporary, disposable copy of a gene, totally unlike the stable, double-stranded DNA, which has to be protected at all costs. So, remember those three-letter triplets we saw in DNA? Well, they have a direct counterpart in RNA, and we call these codons. During transcription, if the DNA triplet is GGC, the complementary RNA codon that gets made is CCG. This is the actual message that the cell's protein-building machinery is going to read. All right, let's head out of the nucleus and onto the factory floor, the cytoplasm. This is where we're going to see how that mRNA message is used to build everything the cell needs, from its basic structure to the thousands of enzymes that make all its chemical reactions happen. Now, to really understand translation, you've got to know the three main players. First, you have messenger RNA, or mRNA. That's the work order, carrying all the instructions. Then you have transfer RNA, or tRNA. Think of it as the delivery truck, bringing in the specific amino acid building blocks. And finally, you have ribosomal RNA, or rRNA, which is a key part of the factory itself, the ribosome, where the protein actually gets assembled. And here you can see the genetic code in action. The ribosome reads these mRNA codons one after the other. It always kicks things off at the start codon, which is AUG. And you'll notice something interesting. There's some redundancy. Proline, for example, has 4 different codons. This actually acts as a nice little buffer against some mutations. And then finally, you have 3 specific codons that act like a period at the end of a sentence. They're these stop signals that end the whole process. So, this is the cellular assembly line broken down into 4 steps.
First, the mRNA work order docks with the ribosome. Then, the tRNA delivery trucks show up. Each one has a complementary anticon that matches the mRNA's codon, and it drops off the correct amino acid. The ribosome then forges a bond, linking these amino acids together, and then it scoots down the line to the next codon, building this protein chain until, wham, it hits a stop signal. Now, a cell can't just have all 25,000 of its genes switched on all the time. That would be absolute chaos and a huge waste of energy. So let's look at the really sophisticated systems that act like a regulatory switchboard, controlling which proteins get made, when they get made, and how many are produced.
The cell basically uses two main strategies to stay in control. Genetic regulation, that's like managing the factory's overall production schedule. It's a slower, long-term approach that decides how many proteins get made in the first place. Enzyme regulation, on the other hand, is like having a manager right on the factory floor who can flip machines on or off instantly. It's a super fast, short-term response that controls the activity of proteins that are already built and ready to go. When you look at transcription, the level of control is just incredible. Every gene has a promoter region, which is kind of like the main on switch, but its activity is tweaked by this whole ecosystem of other elements. You have transcription factors that can either boost or block the process. You have enhancers, which can be far away on the DNA but still crank up the activity. And you have insulators that act like walls, making sure the regulation of one gene doesn't accidentally spill over and affect the gene next door. Okay, let's connect all this back to the real world, to the processes you're going to see every single day in the clinic. We're talking about normal tissue growth, the changes that come with aging, and of course, the uncontrolled cell division that we call cancer. Before a cell divides, it has this monumental task. It has to copy its entire genome, and it has to do it almost perfectly. All of that work culminates in mitosis, this tightly choreographed dance that makes sure each new daughter cell gets an identical and complete copy of the genetic blueprint. You know the little plastic tips on your shoelaces, the ones that keep them from fraying? That is the perfect analogy for telomeres. They are these disposable, repetitive caps on the ends of our chromosomes. Every time DNA gets copied, a tiny piece of the telomere is lost. This is a good thing. It protects the important genetic info from being lost. But it also means there's a limit to how many times a normal cell can divide, a process that's deeply connected to cellular aging. At its very core, cancer is a disease of the genes. It starts when mutations corrupt a normal rulebook for cell growth and division. And these mutations can come from all over, from things in our environment like radiation and chemicals, or even from genes we inherit. But here's the key. It's usually not just one single mutation. It's an accumulation of several hits that finally pushes a cell over the edge into becoming malignant. And this quote from Guyton and Hall, it just nails the two defining traits of a cancer cell. First, it completely ignores the normal signals that tell it to stop growing. The brakes are gone. And second, it often loses its stickiness, its connection to its neighbors. This is what allows it to break free, to wander off, and to invade other tissues, the deadly process we know as metastasis. And this, right here, is the question that's driving the entire future of medicine. With incredible tools like CRISPR that let us directly edit DNA, and with targeted therapies that can shut down the products of those mutated cancer genes, what we call oncogenes, we are finally starting to move beyond just treating symptoms. We are learning how to fix the fundamental errors in the code itself. Understanding this blueprint isn't just an academic exercise. For you, it is the key to the next generation of diagnostics and therapeutics.